So far, we've talked about the first line defenses, which include the skin and the mucous membranes, as well as the things that they make. And we've talked about the second line defenses, which are the defenses inside the body. And that's going to include things like inflammation, fever, macrophages, antimicrobial proteins. And now finally, we're at the point of talking about our defenses that are the adaptive defenses. And these are the defenses that are going to be looking at pathogens that have been able to make it through the other two defenses and then have to learn about them and develop a way to kill them. So in this scenario, where we've got the component of the immune system that is capable of recognizing that something is not supposed to be here, and then crafting an approach specific to that microbe and killing it. So this is why we call our adaptive defenses the specific defense system, because it has the capacity to eliminate almost any pathogen or abnormal cell, no matter how sneaky it may be. Now, among the things that it's going to do, besides just taking out, or not just taking out, but taking out pathogens, is that it's also going to amplify the inflammatory response. Now, at this point, we've learned that just about everything, in addition to doing its thing, is going to amplify the inflammatory response, because we want all the players to show up and cover our bases, because this is a life or death situation for the cells, definitely, and maybe even for the body larger, if this pathogen should be allowed to take hold. The adaptive defenses are going to activate complement, because you might remember when we looked at complement, I said that this is one of those crossover points where we're definitely still in the second line defenses, the non-specific defenses, but we can use antibodies in order to activate complement. So here we are now actually in the adapted immune system, and we're going to be activating complement, which involves the use of that antibody. And the adaptive defense is really very special in its ability to remember the pathogens and launch a good attack. But the weakness about this system is that initially, when it encounters a microbe, it takes a little bit of time for the cells to figure out there's a problem, and then figure out how to deal with it, and then make the cells that are going to make the chemicals to make all of that happen. And that can take up to 2 weeks, 10 days to 2 weeks. So that's the shortcoming of the system. The beauty of the system is that once it's done that, it also makes memory cells that will remember that pathogen, so that the second time it shows up, we launch a very effective, really large immune response, and we can take the microbe out in about 2 or 3 days, and you never knew that it was there because you never even felt any degree of sickness. So that's the beauty of the adaptive immune system. Some important characteristics of the adaptive defenses are that it is specific. So this system looks at a microbe, learns about it, becomes an expert in dealing with it. That's what we mean by specific. Another thing that's important about the adaptive immune response is that it is systemic. It doesn't matter where that microbe is encountered. It doesn't matter if it's in the lungs, if it's in the intestines, if it's in the blood. This system can work anywhere where that microbe is encountered. So that's a really important feature. And another important feature or characteristic about the adaptive defenses is that it has memory. So it remembers the pathogen, and the memory cells linger long after the cells that are designated to fight the pathogen have already passed. And the memory cells are capable of launching a very rapid assault on the pathogen the second time it shows up. So these are 3 really important characteristics to know about the adaptive immune response. Now, the adaptive immune response has 2 primary branches. One is known as humoral immunity. The other one is cellular immunity or cell-mediated immunity. And as the names imply, it basically is telling you where their business is being conducted. Humoral immunity is going to mostly occur in the blood where we release antibodies to float around and attach to antigens and pathogens. And cellular immunity is going to be um, an immune response launched towards cells. So let's have a look at each one of these. When we think about humoral immunity, you need to automatically be thinking about antibodies. That's more or less the basis of humoral immunity. Now, who produces antibodies? Well, it's going to be the B cells, specifically a B cell that is converted into a plasma cell. And these are going to be the cells that are making those antibodies. So what do antibodies do? Well, antibodies are going to bind to a target cell, specifically to the antigenic region of that target cell. And it will temporarily inactivate the cell, which means that it may be preventing it from moving around easily. It may be isolating it and forcing it to stay in a particular state or in a particular area. It can do a number of things. It can prevent it from replicating. So antibodies have the capacity to do a number of things. And in addition to all these other ways that it basically temporarily inactivates the pathogen, the other thing that it's going to be doing with this antibody is marking it for destruction by a phagocyte. So it basically has some way of holding onto it, isolating it, preventing the pathogen from doing whatever it wants to do, and then at the same time acting as that marker for destruction. Humoral immunity has its targets extracellularly. So not body cells, but things outside of the body cells. Things that might be found in the blood, in the interstitial space, or around the cells of the tissues. Those are the targets of humoral immunity. Now, cellular immunity is when our white blood cells, our B cells or T cells, are going to be targeting another cell. And it will attack, for example, a virus-infected cell. That's a form of cellular immunity. It can destroy a cell that might be like a cancer cell or a virus-infected cell, and it can do that indirectly by releasing chemicals that can activate other lymphocytes or macrophages or enhance the inflammatory response. So there's a couple of different approaches that we see in cellular immunity. And it's not that a cell involved in cellular immunity is going to do either one, like either or, directly or indirectly fight something that is problematic in the body. Is that it typically does both of these. So usually these cells have a way of dealing with a problem right there, but then also making sure to call in the reinforcements. So that's kind of a little bit about cellular immunity. And as the name implies, the targets of cellular immunity are going to be cells. Body cells that are infected, body cells that have gone rogue, maybe they're cancer cells. They're cells that need to be eliminated. Now, there's a lot of talk about antigens in the immune system. And we talk about antigens in terms of being substances, molecules found on the surfaces of cells or pathogens that the adaptive immune cells can recognize and that can trigger those cells to mobilize and launch an immune response. Now, the reality is that there's a lot of things that exist on the cells of pathogens, exist on the surface of pathogens and exist on the surface of cells. And they can be identifiers, they can be receptors, and we can even call some of those sorts of surface molecules antigens as well. The distinction really is for us is, does it trigger an immune response or not? So with regard to this particular topic, we're going to look at an antigen as something that has the capacity to launch an immune response. So antigens are really what is triggering the adaptive immune response. So it's not that the pathogen is showing up and that it's the wrong color or that it looks funny. It's that it has that antigen that the immune cell is capable of reading and interpreting as problematic. That's really what the trigger is. So, like I said a moment ago, an antigen is a molecule. Usually it's a very large molecule, often a protein molecule, or at least has a component of a protein in it. And it's going to be something that is not found in the body. So it's going to be something that the cell looks at and goes, that's not something I've ever seen before, because it certainly doesn't belong in here. Because remember, cells, all body cells, um, don't have eyes, they don't have ears, and they have to communicate with the world by their world, particularly by looking at what is on the surface of the other cells that they encounter. So when there is something weird sitting on the surface of another cell, that is a communication to our immune system that we might be having a problem. Now, as I said a moment ago, antigens are going to be these large molecules, typically proteins, found on the surface of cells. And this is a classification of molecules. And the ones that we're concerned about in this unit are the ones that can trigger an immune response. But there's other antigens that exist on the cell surface that don't trigger an immune response, and these end up acting like an identifier. There's a number of different molecules that can act like identifiers, and antigens are some of those molecules as well. And those molecules we would call a self-antigen. Now, the body has taken an inventory in the first few months of life and figured out what's on the surface of all of our cells. And it relays that information to the immune system so that as the immune system starts to launch its activities, it doesn't attack body cells. So our body has a great deal of awareness of what is allowed in this environment of ours, our internal environment, and what is going to be determined to be foreign. And so the immune cells and even other tissue cells are going to be looking at the antigens on the surface of cells and determining whether that is a self-antigen or whether it is a foreign antigen, and then acting accordingly. So it's important for us to understand that we do have self-antigens, and ideally they're not going to trigger an immune response from our cells of immunity. All right. Now, these self-antigens don't just necessarily get stuck up on the the surface of the cell. They have to reside in a special receptor site. And the special receptor site is known as a major histocompatibility complex, and it is it's basically like a receptor, and everybody has a unique um, MHC configuration. So your MHCs are going to be different than mine, and within this receptor, this major histocompatibility complex receptor, is a little groove where a cell can deposit an antigen and display it. Now, what kind of antigen can go there? Well, it might be a type of antigen that can trigger an immune response. And who would want to do that? Well, probably another cell of immunity. So the way that part of the way that the one cell of immunity communicates with another cell of immunity is that it will rip off the antigen of a pathogen and stick it on a major histocompatibility complex on its surface, and then it'll run around and show other cells of immunity what's in the area. So that's one way. It could be that maybe a regular tissue cell is displaying self-antigen there that says, hey, I'm friendly, don't come and take me out. So that's what's happening with our major histocompatibility complex receptors, is that they end up being this place where we can deposit an antigen, whether it's a foreign antigen, and the reason why we would do that is to trigger an immune response, or whether it is self-antigen, which is the cell trying to protect itself from the immune system. So let's talk a little bit about these MHCs and what's going on here. So let's say that we have a cell on the left, and it could be any sort of cell. It could be a body tissue cell, or it could be maybe an immune cell, but it has a major histocompatibility complex, and it has that little groove right here where something can be deposited. So we're gonna run through the scenario of depositing some self-antigen there and some foreign antigen, and then we're gonna see how the immune system, which is represented by this T cell, is going to deal with that. So let's start off by dropping in the foreign antigen. And then what this is going to do, this antigen presenting cell, whether it's a body cell or another immune cell, is going to say to the T cell, hey, come over here and take a look at what I got sitting in my MHC receptor. And so the T cell does. And it takes a look at the first thing it's going to do is read the major histocompatibility complex receptor and make sure it recognizes that. That's the very first thing it's going to do. And then it's going to take a look at that foreign antigen. It's going to go, hey, wait a minute, this is not supposed to be here. And as a result of recognizing the receptor first and the foreign antigen, we get recognition, and it leads to immune activation. And that's the perfect scenario. That's how we want the cells and the immune system to communicate with each other. That's how we want maybe one other cell of immunity to communicate with our T cell and create activation. So let's look at another scenario. So let's say that instead of the foreign antigen, we drop the self antigen in here. So this might be like a body cell that's just trying to protect itself from the immune system and say, hey, my name is, and I'm supposed to be here. So here comes our T cell. And the very first thing that the T cell does is it recognizes the major histocompatibility complex and says, okay, I recognize so far that you are part of me because your MHCs are different than mine, because they're all determined by our genetic code. And so our immune system has to recognize self that way first. And then the next thing is that I'm showing an incomplete union here with the T cell, because what has to happen here is that the T cell says, I know that there's some self-antigen there, but it's not going to trigger a response. So there's not going to be any recognition, and as a result, there will be no activation. And that is ideal when we have a piece of self-antigen sitting in there. So we want the immune system to come and check it out and go, okay, I recognize that you're part of me. Oh, yes, and I see that there's some self-antigen in there, but I'm not going to be triggered by that. So I'm not even going to, pretend I know what it is. So that is the perfect scenario. Let's take a look at a different scenario. So in this case, let's go ahead and drop some self-antigen in there. And now we have, we have a T cell that comes up and says, yes, I recognize the major histocompatibility complex, and I recognize the antigen, the self-antigen, which is a bad scenario. Because if the T cell recognized the cell that was presenting self-antigen, then it would launch an immune attack against that cell. But we don't want the immune system to launch an attack against cells bearing self-antigen. So once this T cell recognizes the self-antigen, the T cell actually gets destroyed. So when T cells are going through their educational process up in the thymus, they're going to be challenged with this. This is a T cell challenge that happens in the thymus gland. And if this T cell recognizes that self-antigen, that T cell doesn't get to graduate from Thymus University. It gets killed on the spot because it's very dangerous to the body. We don't want that guy running around because we're not sure who he's going to kill, but we know it's going to be one of us. Let's look at another scenario, our last scenario. So let's say that we drop in this foreign antigen and we have this T cell, and we can tell already there's a little bit of a problem because when this T cell shows up, it doesn't even make good contact with a major histocompatibility complex. And if it can't recognize that first, then this T cell is useless to the body. So this is a failed recognition, a failed initial recognition. And as a result, this T cell will get destroyed. And this is also another type of a challenge that a T cell at Thymus University has to undergo. And if it fails this test, it'll get destroyed. So now we have a little bit better idea of how the T cell goes about doing its business. Let's talk about the differences between T cells. Now, the thing that we know about T cells, part of the adaptive immune response, and they're going to be the ones that are attacking those intracellular antigens. So we're looking at cells that have been infected with a virus or a bacteria, or maybe some sort of an abnormal cell in the body, something that had bad transmission of information and is doing something weird. A cancerous cell, or in some cases a transplanted cell. So the adaptive immune response is something that when people receive transplanted organs, you always have to be concerned about their activity because they can actually kill the transplanted tissue. Now, when we talk about the different things that T cells are doing, some T cells are going to be the killers. They get out there and they actually do the thing that is destructive to the virus-infected cell or the cancerous cell or whatever's being attacked. While other ones are going to release chemicals that either help regulate the process in some way. So we've actually got 4 different players in our T cell population. So let's kind of make our way into talking about those. And we're going to start off by talking about how they're created. Now, there are 2 general populations of T cells, and we categorize them based upon a receptor that is on their cell surface and what this receptor does. And so we would say that we have got a differentiation of the glycoprotein receptor. Now, one of those receptors is called a CD4 receptor, and the cells that bear that receptor are called CD4 cells. And while the T cell is going through its educational process, it would be classified as a CD4 cell. But eventually, that cell is going to mature, and when it does, it will become a helper T cell. So the helper T is one of the 4 classifications that a T cell can become. It's one of the 4 degree plans that it could pursue while it's at Thymus University. So if it chooses to become a helper T cell, then it has to make sure that it has that CD4 receptor on it and that it gets that in the special moment of its educational process. That's really what's going to happen there. Now, what's the big deal about helper T cells? Well, helper T cells can activate B cells, which we haven't talked about yet. It can activate other T cells, which we also haven't really talked about. It can activate macrophages, which are part of the innate nonspecific defense system, the second line defense, and it can help direct the adaptive immune response in general. Now, some of the cells that enter into the helper T pathway will differentiate further and actually become a regulatory T cell. Now, what's the big deal about a regulatory T cell? Well, the regulatory T cells are the cells that watch the battle play out. They watch the pathogens be attacked, and they watch the immune system start to win the war. And at a certain point, the regulatory T cells say, I think we have waged war long enough, and we need to stop our efforts and just rest for a while and regroup. So the regulatory T cell is kind of like the braking mechanism to this very effective attack that the immune system can launch on a pathogen. All right. The other possibility is that a helper T cell can become a memory T cell. So even though we have a helper T, it can become a regulatory, a few of them will become a regulatory T cell, and some of them will also become memory cells so that we can remember the pathogen that we learned about and launch a more effective immune response later on. Now, that CD4 receptor that we're very interested in when it comes to helper T cells are going to be able to read what is in a major histocompatibility class 2 receptor. Not interested in the class 1s, but is looking for that major histocompatibility complex receptor class 2 and is taking a peek at what's in that. All right, so let's keep moving. Now, the second generalized population, because I said there was 2, the second generalized population of T cells are going to be predicated upon what we call a CD8 receptor. And these cells, these T cells that have a CD8 receptor as they're going through their educational process are going to be called CD8 cells. And when the CD8 T cell matures, it becomes a cytotoxic T cell. And it's these cells that are capable of destroying cells that harbor foreign antigens, whether that antigen is from a pathogen or whether it is from something aberrant in the genome of that cell, because it might be a body cell that just got some bad information in its DNA and is now doing something crazy. All right, so that's that cell, the cytotoxic cell that does the killing. Now, there's a small population of cytotoxic cells, the CD8 cells, that can become regulatory T cells, and there's also a small population that become memory cells. But generally, we would say that your CD4s are going to be your helpers and your CD8s are going to be your cytotoxics, with the understanding that we can get populations of memory or regulatory T cells from either one. Okay. But the majority are going to enter into either the helper pathway or the cytotoxic pathway, because we don't need tons of regulatory or memory T cells floating around. Now, where CD4 cells, the cells with the CD4 receptor, were interested in the major histocompatibility complex class 2 receptor type, the CD8s are going to be interested in the class 1. So that's the difference between these two cells. So basically we've got our bases covered with who can read which of our two major classifications of the MHC receptors. Now, I talked a little bit about how major histocompatibility receptors are going to display an antigen in there. And the question might be, how do we actually get that antigen there? Well, it has to be processed. And we say that it's a processed fragment. So there's something that our cells are doing in order to get that little antigenic nugget to sit in that special groove of our MHC receptors. And we're going to talk about that in just a second. So let's talk a little bit about the class 1 and the class 2 receptors. Where are we going to find these? Well, the class 1 receptor is going to be displayed on all cells that have a nucleus, which means basically all cells except red blood cells. The class 2 MHC receptors are going to be displayed upon the professional antigen presenting cells. So that's going to be our dendritic cells, our macrophages, because we have talked about those a little bit already. And the one that we haven't really talked about is the B cell. But these are the 3 cell types that are phagocytic and are known as professional antigen presenting cells. These are the only cells that bear the class 2 receptor. Okay. Now, I stated this before, I'll state it again. The nature of your receptor is unique to your genetic information, all right? It tells your immune system a little bit about you. Now, it falls within these two categories of category 1 or category 2, but there can be a little bit of variation so that your body knows that it is from you. Okay. So let's take a look at an animation that tells us a little bit more about these MHC receptors and what's going on. We're going to start off by talking about the class 1 receptor. This is the receptor that gets displayed on all cells that have a nucleus, which means everybody but a red blood cell. And you're going to have a CD8 cell that's going to be interested in reading that. So here I have a cell off to the left that has that MHC1 receptor, and then we have our CD8 cell, and I'm specifically saying that this is a naive cytotoxic T cell. So what happens is that our antigenic component gets deposited in that major histocompatibility receptor category 1, and our T cells kind of curious about what's going on in that receptor. So it will come over to the receptor and it will read what's in there, and whatever's going on there will activate that T cell. The T cell will basically give a nod to what's in there. Now, if it's self-antigen, we don't want the T cell to do anything. That's a good response. But if there is a foreign antigen that's in there, then we want our cytotoxic T cell to be activated and then to go on and launch an immune response. Okay. Once our T cell has been activated, we would no longer call it a naive T cell. We would now call it an activated T cell. So we could actually call this one our activated cytotoxic T cell. Okay. So without having been exposed to something that's going to elicit that immune response, we would call these T cells naive. Okay. Let's look at what happens with the major histocompatibility complex 2 type of a receptor. So here we have an MHC2 receptor, and this could be presented on either a dendritic cell or a macrophage or a B cell. Now I have a little asterisk there by B cell because I know we haven't talked about this yet, but when we get to B cells, I'll remind you of this. All right, so in this case, it's not going to be the CD8 receptor on our T cell that can engage with this, but it will be the CD4 that can do that. That's going to lead us to the creation of a helper T cell. So let's say that we have a helper T cell that's never had interaction with a major histocompatibility complex receptor before, and it sees this one there, and so there's going to be a union between the two. And our little naive T cell takes a peek at what's in there, and it's like, oh, so that's what that is. I know what to do with that. And in that way, we have now activated our helper T cell by exposing it to that antigen that was being presented to it in that MHC receptor. Now, a moment ago, I said that T cells are going to recognize process fragments of antigens that are displayed in their major histocompatibility complex receptors. So at this point, the question becomes, how are the antigens processed and how do they get onto that receptor? Well, here we have our pathogen, and we know the story about this little guy. He's got those little X antigens hanging out there, and it's going to stimulate our white blood cell to come along and eat it. So the white blood cell is going to put this guy in a phagosome, and then within the white blood cell, we're going to have a lysosome that's going to dump some enzymes in that phagosome. And the enzymes are going to get dumped in there, and we're going to create the phagolysosome. And the enzymes are going to dissolve the pathogen. And we learned that what we don't need, because the white blood cell is going to sift through the cell parts, the pathogen parts, and what it doesn't need is going to be exocytosed. Yeah. And that's going to leave behind these little antigenic pieces. And so when I said that the cell is going to take what it needs these little antigenic pieces. And guess where these are going to go? These are going to go right up there onto the major histocompatibility complex receptor site. So that's how we process these antigens and get them into that MHC receptor. On this slide, I wanted to put all this information together for you. So we have our CD4 receptor type cells and our CD8 receptor type cells. And our CD4s are going to be naive. They're both naive T cells. Cells that have a CD4 receptor are going to respond to the MHC class 2 receptors, whereas the CD8s will respond to the class 1 major histocompatibility complex receptor. Our cells that have a CD4 receptor on it will become helper T cells, whereas our CD8s will become cytotoxic T cells. And when we look at the activities of these cells, helper T cells are going to help activate other T cells and macrophages and basically fan the flames of the immune response. Whereas our CD8s are going to destroy cells, whether it's an infected cell or a cell with a cancerous cell or some other aberrant body cell. And if we had to compare the two and decide who was the most important, it could easily be said, and has often been said, that the CD4 cells are the ones responsible for the immune response, and that without the helper T cells that CD4 cells become, there would be no immune response. So let's talk about how a T cell has to undergo its educational process in the thymus. So we can say that a fully mature T cell must be able to do two things. First of all, it has to be able to acknowledge and bind to the appropriate major histocompatibility complex receptor, and that is known as positive selection. So it takes a look at that and says, yes, I know who you are. And then the other thing that it has to do is it has to be able to look at what's sitting in that groove of the MHC receptor and above all else, not recognize the self-antigen as problematic. And that is known as negative selection. All right. Now, once our T cell has made it through both positive selection and negative selection, it's going to have to basically choose whether it wants to be a CD4 cell or a CD8 cell. Up until this point, it will have both the receptors on it. Kind of like it's covering its bases. Not sure what it wants to do once it graduates college, so it's just kind of doing a little bit of both in the study world, but there comes a point where it has to finally make a decision and pick a direction. And it will be forced to do that after the negative selection process. So it'll sacrifice one of those receptors and end up with the other one, and then it will be known either as a helper T cell or a cytotoxic T cell. So let's look at an animation about how this process works. So the development of this tolerance, this maturation process for our T cells, all of this is going to happen in the thymus gland. So when we say that we ship off the T cells from the bone marrow off to Thymus University, this is what we're putting them through in order to make sure that when they get released into the general population of body cells, they don't run around and do a bunch of havoc, and they basically protect us. So this is what we have happening here. So this is the process that the T cells have to be able to go through, and the two major exams that they have to pass in order to graduate successfully from Thymus University. And the first one is going to be that positive selection. So what happens here is we take our little naive T cell, and then we have a thymus cell, which is an educator cell. We can think of it as an instructor, a professor. And we want this naive T cell to be able to bind to and acknowledge the major histocompatibility complex. That's the very first thing we want it to do. And when it does that, it passes the positive selection exam. All right. So recognizing the major histocompatibility complex receptor is part one, the positive selection. Now, if at this point the naive T cell fails to recognize that receptor, then that's considered a fail, and that cell will be destroyed. All right. So let's say that we've had our little T cell, our naive T cell, don't know what it's going to be yet, but it has passed the first exam. It's passed positive selection. It can acknowledge and recognize and bind to one of the major histocompatibility complex receptors. So what does it get in return? Well, it gets both a CD4 receptor and a CD8 receptor on its surface. And with those two things in place, it's going to enter into the second most important exam that it needs to pass, and that's called negative selection. Now in this scenario, our little T cell with both its CD4 and its CD8 receptors are going to be exposed to a cell that has some self-antigen in its major histocompatibility receptor site. Okay, and the thing that we don't want to have happen, and that's why I have the red X here, the thing that we don't want to have happen is any sort of acknowledgement or recognition of that self-antigen that will trigger an immune response. So another way that we could say that is that when this cell, our naive T cell, does not bind to self-antigen, that is a pass, because we don't want it to bind to that self-antigen. If it does bind to the self-antigen and it gets stimulated by that, that is a fail, and this T cell will be killed. But let's say that we had a pass. The CD4, CD8 positive cell has passed both the, obviously it's passed the positive selection and it has just passed the negative selection exam, what happens after that? Well, it's either going to end up with the CD4 receptor, in which case we would call it a CD4 positive cell. Some sources would actually say CD4 positive, CD8 negative. Or it's going to end up with the CD8 receptor, in which case we would call it a CD8 positive cell or a CD4 negative CD8 positive cell. And this is a naive T cell that has basically recently gotten its diploma from Thymus University, and it will be released into general circulation. And then once it is activated, it will become what we know that it can become. In the case of the CD4 positive cell, it will become a helper T cell. And in the case of a CD8 positive cell, it will become a cytotoxic T cell. Now, the thing that I didn't include at this phase, mostly because I just ran out of room on the slide, is that we could have a small population of the helper Ts or a small population of the cytotoxic Ts that are going to become those regulatory Ts and those helper Ts. So we just kind of have to understand that a few of those guys are going to end up in these other roles, but the majority of them will either end up as the helper Ts or the cytotoxic Ts. Now, we have talked a lot about all of these different things that we require of our T cells in order for them to do their job. And you might be thinking, man, that just seems like it's so much. I mean, certainly it's a lot to learn, but it's also a lot for the T cell to have to do. But there's a reason why we have that in place. And the reason is that when we activate a T cell, we need to be double, triple dog sure that we want that T cell to do its business, because these are killers. And once they're activated, they might not stop until a regulatory T cell shows up and says, okay, enough already. All right. So it ends up being that we have a number of mechanisms in place to ensure that we are double and checking our activation component. And there is one more we need to talk about as far as this activation, and this is this concept known as co-stimulation. So as I say here on the slide, the T cells are very powerful, they're deadly, and as a result of their capacity to do damage, there is a two-part recognition system that they have to undergo in order to be fully functional, in order for them to be able to act as an immune cell. And I like to think of it as a secret handshake. All right. So let's look at an animation, because you know this is that's what we got going on with this particular lecture. Lots of animations. So here we have an antigen presenting cell, just kind of picking some imagery from previous animations, and we have a cytotoxic T cell that are engaging. And so our cytotoxic T cell has acknowledged the major histocompatibility receptor 1, and it is now checking out that foreign antigen that's sitting in the little groove. But that's not enough to stimulate this cytotoxic T cell. It has to undergo co-stimulation as well. So there's this additional little area here on our presenting cell that the T cell has to kind of do this underhanded handshake to in order to recognize that as well. And when that happens, then our cytotoxic T cell will be fully activated and can go off and do its little immunological function, whatever that is. And in the case of this cytotoxic, it's going to release chemicals and poke holes and fun stuff like that. So the secret handshake, co-stimulation, also has to happen in addition to acknowledging the major histocompatibility complex receptor and successfully acknowledge the foreign antigen. So those are the three things that have to happen in order for the immune response to move forward. Now on this slide, I just kind of recap and give you a real basic explanation of what the 4 different T cells are. So we know that we've got the cytotoxic T cells, which their job is to kill infected cells or cancerous cells. Our helper T cells, which are going to stimulate other T cells, as well as macrophages, and also release chemicals. I didn't put that on here, but you can pretty much assume release inflammatory chemicals and get all that going. But then we have the regulatory T cells, which are going to be the ones that that put the brake on the immune system reaction. Basically, the regulatory T cells say, you know what, I think not only have we won the battle, but we have won the war, and it's time to put up our arms and go home. And then we have what I like to call the scribes, the memory T cells, the ones that wrote the story down and can remember it for the telling on another day. So that when that pathogen shows up again, the memory T cells will be the first ones to recognize it and then launch an immune response, triggering the helper T cells to get the ball rolling and build the army and wage another battle. So we just spent a lot of time talking about T cells, and in some ways T cells are a bit more complicated in their maturation process than B cells are. Although B cells do have their own story, and we kind of need to understand what's happening there. Now, unlike the T cells that get shipped off to Thymus University, the B cells are going to go to a school a little bit closer to home, there in the bone marrow, and that's where they're going to get their education. And the B cells' education is going to be largely predicated on their ability to recognize certain antigens and create antibodies in response to that. So there's a lot of language around how B cells go about doing their business. And we could say that once a B cell has engaged with an antigen, it gets triggered to do this thing called cross-link antigen receptor union. And when that happens, then that's going to lead to clonal selection. So basically the B cell will replicate and proliferate, and then some of those B cells are going to differentiate into effector cells, which are going to be called plasma cells, and those are the cells formerly known as B cells that are going to be responsible for making the antibodies that we're so concerned with in humoral immunity. So like I said, once a B cell has been activated, it is going to differentiate, and most of the B cells are going to differentiate and become the plasma cells, which are going to be making those antibodies that are the basis of humoral immunity. And they're really fast. I mean, they can make these antibodies at a rate of about 2,000 molecules per second, and they can keep that up for almost a week. So once these guys get triggered, they are berserkers, man. They are like going to towns, like get out of my way, I'm making antibodies. And they're going to dump these antibodies into the blood, maybe into the lymph, if that's where this particular B cell is located. And that antibody is going to float around, and it's going to bind to antigens. And once the antibody binds to antigen, it's very much like a Post-it note that says, kill me. That's basically what an antibody is. So the antibody itself doesn't do the destruction. It's the signal that it gives to other cells of immunity, like macrophages, that will show up and actually do the destruction. All right. Now, the clone cells, some clone cells may not become plasma cells, and in that case, these will become memory cells. So yes, we have memory cells that come from our T cells, and we also have memory cells that come from our B cells. And the B cell memories, the B memory cells are doing the exact same thing that or we can think of it that way, that the T cells are doing, which is remembering the pathogen and being available to stimulate the immune system if that pathogen should show up at a later date. So let's take a look at the story of a B cell. So here we have a little B cell, and it's got its little receptors on its surface, and they look, those are the little letter Y looking things. Those are the receptors. And they're later going to become antibodies. Just going to go ahead and say that. That's not true for all antibodies, but it's going to be true for this one. So our B cell comes along and it sees our little foreign antigen, and it's going to allow its little Y's receptors, little Y-shaped receptors, to bind to the antigen on our pathogen. And I want you to note that the receptors are not just staying put on the cell surface, they're actually migrating to the site of the antigen. And this is what we call cross-linking. So a B cell's antigen receptors are going to do everything they can to bind up with a pathogen's antigens. You get a good response that way. All right, so that's what we call cross-linking. Once we have this cross-linking happening, then our B cell is going to proliferate. It's going to make clones of itself. So it's going to say, oh, I've been stimulated. I'm going to make a whole lot of me. And then in the process of this clonal proliferation, some of these B cells are going to differentiate and become plasma cells that are going to make antibodies. And then some of them are going to differentiate and become memory B cells. All right, so this is what happens on the primary response when a B cell encounters a pathogen for the first time. All right, so this is our first response. What happens with our second response? Well, let's take a look at that. Now, before we get into it, let's just kind of review. We know that the primary immune response is when we have the cell proliferation and differentiation upon exposure to that antigen. And I said this earlier in this lecture that this period takes a little bit of time because, first of all, we have to kind of figure out that there's a foreign antigen around, and then we have to figure out how it's put together, and then we have to figure out what sort of cells we need to make in order to take care of it, and then we have to make the antibodies that are going to tag it and then get the immune response that we want out of it. And this is an amazing process, but it's not 100% the first time. The body is learning in this first one. And the lag time between the initial exposure of that antigen to the point where the body is able to launch some sort of an immune response to that antigen can take about 10 days, maybe even as long as 2 weeks. And then after that, the antibodies are going to start to decline. So this 10-day period is kind of that magic number when if you get sick with something, by around day 10, you actually start to feel better. And the reason why is because your body has successfully put together an immune response, made enough antibodies, and started to reduce the pathogen load in your system that's making you sick. All right. So this is the story of our first immune response. Let's take a look at what happens with the second one. So where the first response took a little bit longer to get up and running, the second response, the second exposure to that pathogen is going to give a faster, more prolonged, and much more effective response than what we saw in the first exposure to that pathogen. And the reason why is that we have sensitized memory cells that are responsible for remembering it and can get the message out quicker to get the army started, and let's differentiate into some plasma cells and get this thing taken care of. So this response, unlike the primary response, can happen within hours instead of days. And the antibody levels are going to peak within just 2 to 3 days, whereas the other one was at around day 10, maybe even as long as day 12 or 14. The other thing is the quality of the antibodies is better. These antibodies in the second time around are going to have a greater affinity. That means that they're going to have stronger binding capacity. They're going to be able to latch onto that antigen and not let go. Whereas we might have had just kind of a mediocre attachment to the previous antigen with the antibodies that were made. The other thing that is better about the second response is that the antibody levels can remain higher for longer than what we saw in the first response. So that gives you an extended coverage that we didn't see with our first response. But we do have those memory cells, which can help us out. So in the second response, we're going to have the pathogen, and we're going to have our B cell, and maybe even a memory B cell. Let's say that it was a memory B cell. Do the cross-linking thing, recognize those antigens. But this time, when this sensitized cell engages with the pathogen, and we have the proliferation, we're going to have a huge response with our proliferation. And when we have a huge response with our proliferation, we're actually going to make a lot more of these plasma cells, and those plasma cells are going to make even more antibodies that have that are of better quality and are longer lasting. And this is the beauty of the secondary response. And also, like the first one, we're going to be making some memory B cells. So this is one of the amazing things about the B cell response. It is this secondary response and how it is just so much better the second time around. Cells, they're not all created the same. And B cells are going to have receptors that are specific to certain types of antigens. And they're going to make antibodies that are going to be able to bind to those antigens. So here I'm showing B cells, and we have 3 different receptors, and so the one on the far left is going to bind with our little blue triangles, and as a result of that binding, the sorts of antibodies it's going to make are going to be ones that will have an affinity towards pathogens that have the blue triangle um, uh, antigen on it. And the same thing is true for our little half circle, and it's going to bind up with those, and it'll make antibodies that are specific to that, and as is our little trapezoidal B cell receptor site. So the point of this animation is to show you that, you know, the B cells are generally the same, except the receptors on there are going to dictate which kind of which classification of antigen it will be able to respond to. And then the idea being that once you have that activation, that particular B cell will make the antibodies that are going to be able to latch onto that antigen once that B cell is activated. It'll be able to make the antibodies that are going to latch onto that antigen and then fan the flames of the immune system and get the ball rolling. Now, B cells are amazing things because B cells don't necessarily need a T cell to show up for it to be activated. Now, we might remember that our B cells are known as professional antigen presenting cells, which means that they can do phagocytosis. So let's say that we have our little B cell right here, right there, and then we have a pathogen with an antigen on it. What can happen is that our B cell can go up and it can chow down on that little pathogen, and then it will it'll latch onto the antigen, engulf the pathogen, and then it's going to take that antigen and put it on its major histocompatibility complex. So now it's in a position where it could go introduce a T cell to this to what it has in its major histocompatibility complex receptor, or not. But this B cell has been somewhat sensitized to this antigen. It has a receptor that's built to intercept it. And what can happen is that this B cell on its own, without any sort of T cell intervention, can actually be activated and then differentiate and make a bunch of plasma cells and make a bunch of antibodies that will take care of our pathogen. Now, the problem with a B cell running off and doing this independent of a T cell activation is that there are no memory cells that get made in this process. So there's no memory B cells that get made. All right, so I mean, it's good that we have this capacity, but we don't end up with those memory cells, which means that when that pathogen shows up again, then we kind of have to go through the process all over again. So what we end up with a T cell independent B cell activation is a short-lived immune response with no memory B cells being made. Now, what happens when we have a T cell that gets involved? Well, let's say that we've had our B cell that's already encountered the pathogen, already processed the antigen, and has now displayed it on its major histocompatibility receptor and is presenting it to a helper T cell. So in comes our helper T cell, and it's going to scope out that little antigen, and we got the little co-stimulation secret handshake business going on there. And then once that happens, the T cell is going to release some chemicals that are not only going to encourage the B cell to proliferate and differentiate, so we're going to get those plasma cells with their antibodies, but we're now also going to get differentiation into a memory B cell. So with T cell dependent B cell activation, we get a longer lived immune response, and the reason why is because we end up getting those memory B cells. So humoral immunity is all about the antibodies. So what about the antibodies? Well, antibodies are also known as immunoglobulins, or the Igs, and these are largely proteins, and these are made by those activated B cells that we now call plasma cells, and they make up the gamma globulin part of our blood. So you might remember when we were looking at the plasma proteins back in an earlier unit, we said that actually most of the plasma proteins are made by the liver, except for the gammas, and they were made by the activated B cells, the plasma cells. Now, the thing about the antibodies is that they're capable of binding specifically with antigens that are detected by those B cells. And they don't actually do the destruction. And a lot of people think that that's the case, but they don't. Like I said earlier, they're like a post-it note that gets attached to something that is bearing that particular antigen that the antibody can respond to. And it's a post-it note that says, "Kill me." So it stimulates somebody else to show up and do the dirty deed. But that's how the system works. When we talk about antibodies, we group them into 5 classes of immunoglobulins. So let's take a look at those. Now, when we draw these antibodies out, you'll see them drawn either as a Y-shaped structure or sometimes a T-shaped structure, but the bottom line is that they have a stem, and off that stem up at the top there are 2 branches. And it depends on the type of antibody and how it gets drawn, whether those branches look more like a T or more like a Y. But we would call this overall Y, and I'm going to go with a Y-shaped structure, the antibody monomer. So each one of these proteins can either act on its own, or like proteins can come together with other monomers to create something that will create an action as a unit. So that's not uncommon behavior of a protein. Basically, it can either act on its own as a single tertiary structure, or it can become part of a quaternary structure and then become active that way. Now, when we look at this Y-shaped structure, it has 4 chains, and there are going to be 2 identical heavy chains, which are somewhat hinged in the middle, and then there's going to be 2 identical light chains. Now, there's going to be a variable region at each of the arms of our Y structure, and these are going to be the areas where the antigens are going to bind. When the antibody makes contact with the antigen. So it doesn't bind the antigen at the stem, it binds it at the tips of the arms where the variable regions are. And the variable regions allow this antibody to be specific to that antigen. So I don't go into a lot of detail about these images, but I will go into a little bit of detail about this one. So what we see here is that we've got, our 2 arms right here, and then we have our stem right here, and we have this hinged region right through there. And then this is a chain, protein chain. Here's another one, and then here's a complementary on this side, and then complementary on that side. And these are bound together by these sulfide bonds. Just going to let you know what that is. It's a disulfide bond. And it's at the tips of these these branches here that we're going to have the variable regions. All right. So we've got a variable region right here, variable region of the heavy chain, and we have a variable region of the light chain. And it's in this area where the antigen will bind. The rest of it, largely the same, antibody to antibody. So as we just saw, we've got heavy chains and light chains, which are the proteins that are making up the basic structure. And the difference between each antibody, it has to do with due to differences in the heavy chains. That's really what we're seeing, is a difference in the heavy chains. The 5 primary classes of antibodies are going to be your IgMs, your IgAs, your IgDs, IgG, and IgE. And we're going to take a look at each one of these. So we're going to start off by talking about the IgM. This is a huge polymer that's made up of, let's just count them, 1, 2, 3, 4, 5 of our Y-shaped structures. All right, so it's a pentamer made up of 5 of our little Y-shaped structures. This is the first antibody that gets released when we have an encounter with an antigen for the first time, this guy. Right. This is a potent agglutinating agent, which means it has the ability to bind to multiple pathogens or multiple antigens and create these little islands of gummed up antibodies and antigens and pathogens all kind of hanging out together in a clump. We saw something like this when we were talking about blood typing, and we talked about the agglutination reaction. Remember that? So that's what we're talking about. It readily fixes and activates complement. So yeah, we love that. So it helps that whole cascade of launching the Big Mac attack that kills a pathogen. All right, so let's talk about the IgA. Now the IgA is a dimer. So we've got two of our Y-shaped structures. There's one on this end, and then there's another one on this end. And this makes up a dimer. The word there, di, means two. And this is something that we're going to find in mucus and in other secretions. And it's almost, we could almost say that it's strongly associated with our first line barriers because, you know, mucous membranes are a first line barrier. So even though this is an adaptive defense, it's humoral defense, which makes it a third line defense, it's strongly associated with structures of the first line defenses. Just wanted to point that out. And because it is there at the first line, it helps prevent the entry of pathogens. So our IgAs can actually help stop a pathogen from gaining entry into the body tissues, largely because it's located in a first line barrier. Now, IgD is a monomer, so it's just going to be one of those Y-shaped proteins, and this is going to be the one that actually attaches to the surface of the B cells. And these are going to be the ones that function as B cell receptors. Now, IgG, this is going to be most of the antibodies that we're going to find in plasma. And these are going to show up in secondary and late primary responses. So it's our IgM that shows up in the most abundance first in a primary exposure. And then after those peak, after the IgMs peak in that primary exposure, then we're going to see the IgG population increase. And then in the second exposure, it'll be the IgG that's the big player there. All right. I tend to think of the IgGs as the 911 of the antibodies. Now, IgG is also unique in that it is the antibody that has the capacity to cross the placental barrier. The other ones don't, but this one does. And then our last one is the IgE, and this one is often involved in allergies and parasitic infections. And one of the things that it does is it causes mast cells and basophils to release histamine. And you might remember from the blood unit that mast cells and basophils are very similar. In fact, it used to be believed that they were the same cell, except one was in general circulation and one was residing within tissues. But we now know that they are histologically different cells. Basophils are the ones that run around and move around freely in the body, and the mast cells are the ones that take up residence in the tissue and live out their lives there. Now, they're both loaded with histamine, and they kind of behave the same under similar circumstances. And for these reasons, they were initially thought of as the same cell. But as we just determined, they are something different. Now, the way I remember IgE is that we had a white blood cell called eosinophils that were dedicated to allergies and those parasitic worms. And so I remember IgE is kind of similar to eosinophil, the E thing going on there. So hopefully that helps you too. All right, let's talk about how we get immunity. And there's a number of ways that we can do that. And we classify our immunity as either an active immune response or a passive immune response. And within each one of those categorizations, active and passive, we can also have natural immunity or artificial immunity. So we can have active natural or active artificial, or passive natural and passive artificial. So we can mix up all of these names. So let's take a look at each one of these and see what we can figure out about them. Now, active humoral immunity basically means that you were exposed to a pathogen and your body now has the antibodies capable of taking that pathogen out. So you'll always have the creation of the memory cells, and you'll be able to create the plasma cells from your B cell clones. So you'll have all of that in place forever and ever. All right. So how were you exposed to that pathogen? That's going to lead us down the pathway of either it being naturally acquired immunity or passively acquired immunity. Now, if it was naturally acquired, you got sick. The pathogen made its way in and your body did the immune response that we've been talking about for the entirety of this lecture. So that is what we would call active natural immunity. Now, you might have been injected with a weakened strain or population of the pathogen by way of a vaccine. And when that happens, when you have a medical intervention, that's no longer natural. We call that artificial. Now, your body still had to go through the process of responding to that antigen and that pathogen, but we basically stacked the odds in your favor so that you could have the immune response without actually getting sick. So that's the beauty of a vaccine. So when there's a medical intervention, it's considered artificial. And because your body had to make the antibodies, it's still active. So this is a scenario of active, artificial, acquired immunity. Now let's have a look at the passives. Now, in active, our body was actually making the antibodies in order to deal with what was being placed in the body, whether naturally or artificially. But in the case of passive humoral immunity, what's going to happen here is that your body will be handed the antibodies and your B cells will not be challenged and your B cells will not make antibodies and your B cells, as a consequence, will also not make any memory cells, which means that after this episode is done, your body will have no memory that it ever occurred. Now, why would we give someone passive humoral immunity? Well, there's a couple of scenarios. Maybe your body just doesn't have the capacity to defend itself at that moment. That could be a reason. Maybe the pathogen that you were exposed to can work faster than your B cells can to deal with it, such as in the case of a rabies infection. Your body will not respond to the rabies infection fast enough to save your life. So then that requires medical intervention. So let's actually look at the slide where we go through all of this and get all that squared away. All right, so we're on the slide with our passive immunity, and we have a couple of scenarios where we can have natural passive immunity, and that's going to be when the antibodies are conveyed to a fetus or a baby from the mother. And that's going to happen either through the placenta or through mother's milk. So those are your two examples of naturally acquired passive immunity. Now, this is the scenario where the fetus's immune system has not been, is not in place, not fully functioning. So the fetus and the newborn are relying upon the mother's immune system to provide some degree of protection. Now, as I stated on the previous slide, artificially acquired passive immunity is going to be a scenario where you're going to be given the antibodies because time is of the essence. Usually that's the scenario. Time is of the essence. So what happens there is that you're just given the antibodies that the doctors know are going to deal with the antigen that has been that has gained entrance into your body. And so, like I said, the rabies vaccine is a really good example of that. Rabies takes hold really, really quickly. And if you don't get that taken care of pretty fast, then chances are, well, your body will not outpace the rabies infection in time to save your life, which is why we go ahead and give a vaccine for that. All right, so let's take a spin by the homeostatic imbalances before we wrap up this unit or this entire chapter. Now, autoimmune disorders are kind of a tragic situation because what's happened is that the tolerances have been confused or have broken down such that our T cells are now starting to recognize self-antigens as problematic. And we're developing antibodies in some cases that can attack our tissue cells, and then off we go. Now, we don't always know why that's the case. We're gaining better understanding about what's happening, but there is some speculation that there are environmental factors such as contaminants that get into the body, and they're antigenic, so they're creating a response, but then they're close enough to body cells that the immune system gets a little bit confused. So there's that sort of scenario that can happen. And there is some speculation that there's a genetic component. I know that a lot of times science will say, oh, well, there is no genetic component, or there is, they make those determinations. But I say that we get our physiology from somewhere. And it's typically going to either come from the mama or the daddy. So look at your parents and see what their tolerances are, and that probably is going to give you a pretty good clue about what yours are. Now, how do we deal with this once we're into this sort of scenario? Well, it really depends upon how bad that immune disorder is. You might be able to manage it with an anti-inflammatory protocol, which can include an anti-inflammatory diet, and also taking a look at things like food sensitivities, environmental sensitivities. So that means you'll have to have some blood testing done to see what's going on, and then eliminate those things from your environment. Stop poking the bear of the inflammatory response. So yeah, there is that approach, and then of course, making sure that you're giving your body things that it needs to rebuild itself. That's the other part of that protocol that's not here on this slide. If your condition is really, really bad, then they might end up giving you immunosuppressive drugs. So rheumatoid arthritis might be one of those situations where you're going down the path of the immunosuppressive drugs. So that's a little bit about immune disorders in general. So what are some examples of autoimmune disorders? Well, rheumatoid arthritis. And rheumatoid arthritis is a tragic scenario where the cartilage, joint cartilage, is going to be damaged to the immune system. Now, that's not the only thing that can happen with rheumatoid arthritis. A number of tissues can become involved with that. But the longer we have these autoimmune disorders, and the more science is looking at them, the more we're starting to see that there is some crossover in virtually all of these, where it's not just one tissue that gets targeted. Eventually, multiple tissues will be targeted, and the patient will develop multiple disorders over time. So if this is your patient, if this is you, if this is your family member, it's really important that you be proactive in dealing with this and eliminating the offending agents and dealing with the inflammatory response that's happening in your body, because one thing will lead to another. So that's never good news. Lupus is another one, and in lupus, this one you're going to see a lot of different tissues involved. It's not just pointing your finger at one. The hallmark presentation with a lupus patient is that butterfly rash across the cheeks of the face. But they can have a lot of different tissues that get affected with lupus. Celiac disease, this is your patient that's going to have intestinal issues due to the presence of gluten. They can't digest gluten, and it triggers an autoimmune response. And the same thing is true of people who have a gluten allergy. But there's a difference in celiac disease and gluten sensitivity or a gluten allergy. One is a genetic issue, and the other one is kind of like you develop the allergy over time because of a health issue that was underlying the allergy that causes the intestinal damage. But they're very similar, and you would more or less treat them the same way. Hashimoto's disease is when the immune system decides that your thyroid's a problem. And believe it or not, there's a really strong tie-in with gluten and Hashimoto's disease. So gluten allergy can lead to Hashimoto's disease, or gluten allergy can exacerbate Hashimoto's disease. And the reason there is that the gluten protein mimics some of the tissue that we find in the thyroid. So if you have Hashimoto's disease and you continue to eat gluten, you can actually keep the immune system highly stimulated, and the more that the immune system is stimulated, the more it will attack the thyroid. It's going to attack everything, but the more it's going to attack the thyroid, then the worse your condition will be. So there's a lot of crossover with these. All right. Now, the way that I would approach learning the autoimmune disorders that we want you to learn is to basically find the thing that you can remember about that particular disease. Because each one has basically a single identifier that can carry you back to that disease. So Hashimoto's is as a thyroid, rheumatoid arthritis is cartilage, celiac is the gut. That's more or less how I would try to make those associations. Now, in the previous slide, I did use the words allergy and sensitivity. And what's happening here is that this is when the body undergoes an inflammatory response to something that is typically thought to be non-antigenic. Something that shouldn't be creating that inflammatory response, something that's normally found in nature that most people aren't going to respond to, such as pollen or dander or gluten, for example, food allergies. So this is somebody's body that has just decided that now this is a problem. And what happens here is you get a big activation of IgE and IgG. So if you go and you have food allergy testing done, they do the blood draw, not the skin prick, but the blood draw. They're going to be looking at your body's response, the IgG and the IgE response to like a couple of hundred different known food allergy or foods that create allergies, and they'll help you figure out where you fall on the spectrum of things. So yeah, allergies and hypersensitivities are when somebody's immune system is now reacting to things that are typically thought of as non-antigenic. Now, when it comes to lymphomas, this is going to be a cancer that involves lymphatic tissue, and it's an overgrowth of white blood cells. And we're going to see tissues that are affected, including the lymph nodes, the spleen, the thymus, and the bone marrow. And there's two general types. You have your non-Hodgkin lymphomas, which is the most common, and then you have your Hodgkin's lymphoma. So basically, if it is not a Hodgkin's lymphoma, it's classified as a non-Hodgkin's lymphoma. Lymphadenopathy is when you have an inflamed lymph node, and there can be many causes for this. It might be that your body's waging an infection in that lymph node. That'll cause it to inflame. Maybe there's cancer in the area. That can also create an inflamed lymph node. Or there might be some sort of an autoimmune disorder and the body's undergoing that sort of response. The cause of the inflamed lymph node is going to determine the treatment that is involved. And the treatment may be simply deal with the infection, deal with the immune issue, or removal of the lymph nodes. Mononucleosis, this is a highly communicable disease, and it's caused mostly by the Epstein-Barr virus. When you have a person coming in with this, they're going to have extreme fatigue. They'll have a sore throat. They'll be running a fever. They'll have swollen lymph nodes in the neck, in the armpits, and they might even have a swollen spleen or a swollen liver or both. And there's going to be some sort of a rash on the skin. This is diagnosed with a blood test. And the good news is that it typically resolves on its own, and you basically just treat whatever discomfort with an over-the-counter medication. The thing about mononucleosis is that if you do have splenomegaly, the swollen spleen, or hepatomegaly, the swollen liver, you don't want to be real active, because if you're out there running track or climbing on the monkey bars or swinging from the tree and whatever activities that you might be doing, when you have those two organs that are swollen, you run the risk of rupturing them. So often a patient who has a case of mono is told to take it easy and not participate in very athletic endeavors or do a lot of physical activity, because you want to just you want to give the body time to rest. Number one, you're probably not going to feel like it. Number two, and number three, you don't want to run the risk of accidentally rupturing your enlarged liver or your enlarged spleen. Now, for my Galen students, there is an extensive component of the homeostatic imbalances in the module that is dedicated to talking about HIV and AIDS. So we'll touch on it here. So HIV is the human immunodeficiency virus, and it has transmission routes that involve basically the exchange of body fluids. Body fluids can include blood, vaginal fluids, semen, and pre-ejaculate. Its basic target are the helper T cells in the body, but it can also include macrophages, dendritic cells, and remember, your macrophages and your dendritic cells are going to be your antigen-presenting cells, which work closely with the helper T cells. So basically what HIV does is it goes in and it wipes out the body's immune response. That's what makes it so deadly. The way that this is diagnosed is through blood testing. So you can diagnose the presence of the virus early on. Now, if the virus is allowed to do its thing in the body for long enough, then you develop what's known as AIDS, which is the acquired immunodeficiency syndrome associated with having had the immune system decimated. So what has happened in the AIDS patient is that we've seen the destruction of helper T cells, macrophages, and dendritic cells. And this leaves the body open to a plethora of opportunistic diseases. And what we mean by opportunistic diseases are diseases that typically are not ever manifested in a reasonably healthy person. But like I said, because this person has no helper Ts or the majority of the APCs, the antigen presenting cells, then we don't get the immune response to deal with these things that are taking hold. Now, when a patient has AIDS, the initial response, or even when they have the initial HIV infection, the initial response can be very similar to flu-like symptoms. And then there's going to be this latency period where it doesn't seem like anything's going on. And people early on in the infection, people thought that they had gotten over it. And this is before we knew what was actually happening with AIDS. This is back in the 80s when HIV was first discovered. During the latency period, most patients will develop lymphadenopathy, and so there's these swollen lymph nodes. They're not really sure what's going on, and then the next thing you know, they started having all these. I'm kind of giving you the history here. The next thing you know, that there was night sweats, and then opportunistic diseases, and then finally the scientists took a closer look and figured out what was happening. So in this patient who has the flu-like symptoms and then has a latency period, will then develop some swollen lymph nodes at some point, maybe not have other symptoms, maybe have some night sweats. But meanwhile, what's happening is the immune response is being decimated. And at the point that the patient has a CD4 helper T cell, we're going to call it the CD4 positive T cell count, below 200 cells per microliter of blood or cubic millimeter of blood, that's when we diagnose that patient with full-blown AIDS. So basically, that's the point at which medicine has decided that the immune system is no longer capable of defending the patient. And with that, we are done. Thanks for watching.